bacterial conjugation, including both F plus F minus transfer and HFR-mediated recombination. Each question includes 5 choices and full rationale for correct and incorrect answers. Question 1, basic conjugation F plus FX. A researcher is studying horizontal gene transfer in bacteria. She mixes an F plus strain of Escherichia coli with an F strain. After incubation, she finds that the recipient F strain now has the ability to form a sex pilus. What is the most likely explanation? A. Transfer of chromosomal genes by bacteriophage. B. Uptake of naked DNA from the environment. C. Direct plasmid transfer through conjugation. D. Point mutation leading to sex pilus gene activation. E. Integration of a lysogenic phage. Correct answer. C. Direct plasmid transfer through conjugation. Explanation. The F plus plasmid contains genes for the sex pilus and conjugation. In F plus F conjugation, a single strand of the F plasmid is transferred to the F cell, converting it into F plus. A. Describes transduction. B. Describes transformation. D. No mutation occurred. This was horizontal gene transfer. E. Lysogenic phages may integrate DNA but do not transfer sex pilus genes. Question 2. Conjugation outcome, F plus F. Which of the following is the most likely outcome when a typical F plus bacterium conjugates with an F bacterium? A. The F bacterium becomes an HFR cell. B. The F bacterium becomes F plus. C. The F plus bacterium loses its fertility plasmid. D. The F bacterium integrates viral DNA. E. The F plus bacterium becomes diploid. Correct answer, B. The F bacterium becomes F plus. Explanation. In classic F plus F conjugation, the F plasmid is transferred, converting the F recipient into an F plus donor capable of future conjugation. A. FR forms when F plasmid integrates into the chromosome. C. Donor retains its own copy. D. This is transduction. E. Bacteria are haploid. Question 3. HFR-F conjugation. A high-frequency recombination, HFR strain of E. coli, is allowed to conjugate with an F strain. Afterward, the F cell does not become F+, but acquires some chromosomal genes. What explains this result? A. The entire F plasmid was transferred. B. The plasmid was degraded in the F cell. C. Chromosomal genes flanking the integrated plasmid were transferred. D. The recipient cell underwent transformation. E. The sex pilus did not form. Correct answer C. Chromosomal genes flanking the integrated plasmid were transferred. Explanation. In HFR-F conjugation, a portion of the F plasmid and adjacent chromosomal genes are transferred. Since the entire F plasmid is rarely transferred, the recipient remains F but gains new chromosomal genes. A. Entire plasmid not transferred, conjugation often interrupted. B. Degradation would prevent gene acquisition. D. Not transformation, this is conjugation. E. Sex pilus formed, conjugation occurred. Question 4. Experimental design, DNA sensitivity. In a lab experiment, 2 bacterial strains are mixed, one F plus and one F plas. The researcher adds DNase to the culture. After conjugation, many F cells are converted to F plus phenotype. What conclusion can be drawn? A. The mechanism is transduction. B. The DNA transfer occurred via transformation. C. Conjugation occurred and DNase had no effect. D. F cells mutated spontaneously. E. The F plasmid was transferred by a bacteriophage. C. Conjugation occurred and DNase had no effect. Explanation. Conjugation involves protected DNA transfer through a mating bridge. So DNase, which degrades free DNA, has no effect. This distinguishes conjugation from transformation, which is DNase sensitive. A, B, E. DNase affects transformation, not conjugation or transduction. Spontaneous mutation is unlikely in all F cells.